So this is a pulse oximeter. You may have seen this at the doctor's office before. It goes on your finger, right on your fingertip like that, and it measures your blood oxygenation levels. When you breathe, oxygen enters your body through the nose and goes into your lungs. From there, it enters your blood and attaches to proteins in your blood called hemoglobin. Then, the newly oxygenated blood moves through the rest of your body and brings oxygen to all the tissues. Here's how it works. Put it on the fingertip, turn it on, wait for it to read, and 97% of his hemoglobin molecules are carrying oxygen.
While pulse oximeters are readily available and easy for everyone to use, they are limited to measuring blood oxygen saturation, so they cannot tell us how oxygen is used by the tissue. In addition, there are factors that affect their accuracy, such as poor blood circulation, movement, skin thickness and temperature, etc. For these reasons, we need additional techniques that can measure directly how much oxygen is in tissues such as the skin.
Please follow me to the lab. Welcome to the Evans lab. Instead of measuring how much oxygen is carried by the blood, our lab is developing techniques to directly measure oxygenation of the skin using new glowing molecules inside materials that can be shaped into different forms. Here is a material that's shaped into a flexible ring that changes color to indicate oxygenation changes when we shine blue light on it. It contains a molecule that always glows green and a molecule that does not glow when there is sufficient oxygen but changes to bright red when there is no oxygen present. So you can see here that we have attached this oxygen sensing ring onto the finger of a glove, and the glove is connected to a tube that we can use to run different gases to simulate the different oxygen levels that your blood would deliver to the tissue under different conditions. The ring glows when we shine blue light on it, and you can see now that there is normal oxygenation, so the ring is glowing green. You can see here, this is the air tank, and I'm already running air flow to simulate normal oxygenation. When I close this valve and turn on the next valve, that's on the nitrogen tank, the nitrogen will displace all the oxygen and it'll simulate a condition known as tissue hypoxia, and the oxygen sensing molecule will start glowing brighter and brighter red, and eventually it'll become visually brighter than the green.
Once a device has been tested in the lab, we can start testing it in people. For the oxygen sensing ring, we can use a device that gives us the same or similar results, like pulse oximeter. Then we can compare the results and see if they agree. In order to test in people, we can use a population called healthy volunteers, which are just regular folks who want to help us with science. In order to do this, we have to bring our device description, a protocol, and how we're going to test it to a committee called an Institutional Review Board, or IRB. This committee reviews our plan and makes sure that it's safe to test in our volunteers. This is the first step to getting any device into a doctor's hands.